Hi viewers, welcome back to Mani's Microbiology. In this video, I would like to explain general properties and structure of viruses. Historically, two scientists contributed to the discovery of first virus, tobacco mosaic virus. Ivanovsky reported in 1892 that extracts from infected leaves were still infectious after filtration through a Chamberlain filter candle. Bacteria are retained by such filters. A new world was discovered, that is, filterable pathogens. However, Ivanovsky probably did not grasp the full meaning of his discovery. Martinus William Beijerinck in 1898 was the first to call virus the incident of tobacco mosaic. He showed that the incident was able to migrate in an agar gel, therefore being an infectious soluble agent or a contagium vivum fluidum, and definitely not a contagium fixum as would be a bacteria. Ivanovsky and Beijerinck brought unequal but decisive and complementary contributions to the discovery of viruses. Since then, discoveries made on tobacco mosaic virus have stood as milestone of virology history. Beijerinck published his findings, calling his discovery a contagium vivum fluidum. He is often called the father of virology. Between 1904 to 1908, V. Ellerman, O. Bang, H. Valley, and H. Carey, they first demonstrated a human leukemia causing virus retrovirus, that is the first demonstrated human or animal virus. Viruses cannot be considered as a living being or a non-living being, although they may seem to behave as living things. Viruses are actually acellular, non-living particles. Viruses are not considered to be living organisms because they are incapable of carrying out all life processes. They are obligate intracellular parasites. They depend on biochemical machinery of the host cell for replication, and they are not classified within any kingdom. Viruses cannot able to reproduce on their own like binary fission. They infect a host cell and uses the host's replication process to produce identical progeny virus particles. They infect organisms as diverse as bacteria, plants, and animals and exist in netherworld between a living organism and a non-living entity. Unlike living things, viruses replicate, grow, metabolize, and reproduces, but they entirely depend on the host cells. Viruses are sometimes called virions. A virion is a complete virus free in the environment, not in the host. A virion consists of at least a nucleic acid core and a outer protein coating or capsid. Sometimes a virus will have an outer envelope made of protein and phospholipid membranes derived from the host cell. The viral genome may be an RNA or a DNA, but not both. The genetic material is packed inside a protective shell of protein called capsid. The virion particle can be measured using nanometers. Clinically important smallest virus starts from 18 nanometer, that is parvovirus, to the largest virus of about 900 nanometer, that is Ebola virus. It requires an electron microscope to visualize the structure of virion. Structurally, larger virion can hold a larger amount of genome that can encode more amount of proteins to survive inside the host and infect the host cell, and they are also considered to be more complex to understand. Virus can infect all life forms starting from bacteria, fungi, protozoans, plants, and largest animal on Earth. Regarding the structure of virus, the viral particles are simple, microscopic, and minute in nature. It is even smaller than the bacterial cells. A simplest virion may contain a nucleic acid packed inside a protein coat or capsid, or it may also contain a membrane, that is envelope. Some of the virions may also contain certain essential or accessory enzymes or other protein to facilitate initial replication in the cell. Especially RNA viruses requires these enzymes for initial replication inside the host. The capsid or nucleic acid binding protein associate with the genome to form a nucleocapsid. Inside the virus, the genome may be of either a DNA or an RNA. The DNA can be present as a single-stranded or a double-stranded, and it may be formed in a linear manner or in a circularized form. The RNA may be present in a single-stranded form or in a double-stranded form. In single-stranded, it may either present as a positive sense RNA, that is, it may directly perform as an mRNA or messenger RNA. In the negative sense form, it requires a complementary strand to act as an mRNA. In some of the viral genome, it may also contain a reverse transcriptase enzyme for replication inside the host cell. The RNA genome may be present in a segmented form, that is in a several pieces. Each piece may have a function or it encode one or more genes required for the viral particle. The capsid and envelope are very much important structures for package and protection of genetic material of virus, and they also act as delivery vehicle during the transmission of virus from one host to another host. And they are most importantly involved in tissue tropism, that is a spread of virus within the host to the target cell. The viral attachment protein present in the surface of capsid and envelope mediates the attachment of virus over the target cell. After the delivery of genetic material, the outer coverings, capsid and envelope, becomes inactive and they are unable to cause any infection. Simultaneously, antibodies were generated against these outer structures, which helps in prevention of the viral infection in the host. This is a common icosahedron structure of a virus, which consists of double-stranded DNA as genetic material surrounded by a nucleocapsid and a lipid envelope layer. In between the lipid envelope layer and the nucleocapsid, there is a space which we call it as tegument, consists of essential proteins and enzymes for viral replication. The lipid envelope consists of envelope proteins made of glycoproteins, which are all responsible for the virus attachment to the host receptors. The functions of capsid. The capsid is an important rigid structure. It has the ability to withstand harsh environmental conditions. The properties of capsid are: it protects the viral genetic material from the following environmental conditions and make it stable. The environmental conditions are temperature variation, acid environment, proteases, detergents, and a drying environment. It is released from cell by lysis. And it also has several consequences in human. It can easily spread through fomites from hand to hand, by dust and by small droplets. It can get dried out and retain its infectivity. It can survive under the adverse conditions of the gut of human. It can be resistant to detergents and poor sewage treatment. It can be destroyed by antibody and immune protection cells of host. Capsomeres. The protomers and capsomeres are the individual structural proteins associated into subunits, progressively larger units that forms the capsid. The protein and its bonds are the components of capsid that have chemical features that allow them to fit together and to assemble into a larger unit. The symmetric nature of viruses brought about by the arrangement of capsomeres into two structures, that is helical and icosahedral structures. The helical structures appears as rods, whereas the icosahedron structures appears as an spear shape. The envelope is an important structure of the virus. The envelope, it is made of lipids, proteins and glycoproteins of host. It requires to be maintained in aqueous solutions. It gets readily disrupted by drying alcohol, acidic conditions, detergents and solvents such as ether, which results in inactivation of the virus. As a result, enveloped viruses must remain wet and are generally transmitted in fluids, respiratory droplets, blood and tissue. The enveloped viruses most commonly cannot able to survive the harsh conditions of gastrointestinal tract. All of the negative stranded RNA viruses are enveloped. Viruses are commonly observed in two different symmetry. The first one is the helical symmetry. The protein subunits and the nucleic acids are arranged in a helix or spiral tube-like structure. The capsomere present in the capsid are coiled around over a central axis to form this helical structure. Most of the helical viruses are enveloped in nature and they belong to the RNA viruses. The tobacco mosaic virus is an example of helical viruses. The second one, icosahedral symmetry, where the protein subunits assemble into a symmetric shell that covers the nucleic acid containing core. An icosahedral symmetry is a polygon with 12 vertices, 20 facets and 30 edges. Each facet is an equilateral triangle where the viral proteins remain. The icosahedral capsid is the most stable and found in human pathogenic viruses, namely adenoviruses, picornaviruses, papovaviruses, herpes viruses, and many more. Thank you for patient listening. Please watch, share, and subscribe to my YouTube channel. Thank you.